My name is Judy Bai, and my project is on computational prediction of COVID-19 risky genes associated with lung cancer. So a brief little introduction. Lung cancer is an uncontrolled division of faulty cells happening in the lungs, and unfortunately, it is the third most common cancer in the United States. COVID-19 is a recent virus involved in the global pandemic causing viral lung infections. So just recently, I have noticed that lung cancer patients who have COVID-19 experience more severe symptoms of the virus compared to the normal population. Along with that, not many studies have been conducted about any genes that could be connecting the two diseases. In result, I use bioinformatics methods to identify any candidate genes that are contributing to lung cancer. This leads to my hypothesis, which is genes that are differentially expressed in lung cancer should have the same expression direction in COVID-19 cases. Therefore, they are candidate genes contributing to the co-occurrence of lung cancer and the coronavirus disease. These are all the databases I use to conduct my project and how I use them. First of all, I searched for candidate genes with certain expressions for lung adenocarcinoma, also called LUAD, using the Cancer Genome Atlas. Then I did protein-protein interaction with the string database to find any connections between the candidate genes. Next, I conducted pathway analysis and functional annotation for the selected candidate genes that were downregulated using the David database. I then did an identification of conserved domains for downregulated genes in LUAD, and using the Kaplan-Meier plotter, I calculated the survival rates of lung cancer patients with the downregulated genes. Last but not least, I cross-checked for the downregulated genes expressed in LUAD and also using infection literatures. So this figure was made from the protein-protein interaction of string, and looking at it, I noticed that the genes involved in basic lung functions and immune deficiency had a connection. More specifically, they interacted closely. Another thing I discovered was that the genes with the close protein-protein interaction from the last slide had a role in immune functions, and this was from doing the functional annotation analysis. These are the results of the Kaplan-Meier plotter. So after analyzing the 8 graphs, I saw that 22 out of 25 patients who were expressing most of the downregulated genes with higher expressions in normal conditions did survive longer. After conducting the last step of my project, which is cross-checking for downregulated genes, I noticed that the low viral cases had several of our identified surfactant genes, and those would be SFTP, SFTPB, SFTPC, SFTPA1, and SFTPA2. And those were enriched compared to the high viral cases. So looking over all of my findings, I can conclude that the reason why lung cancer patients have more severe symptoms than the normal population is because of the surfactant genes. Lung cancer patients' surfactant genes are very underexpressed in result of the cancer, and this makes it easier of a task for COVID-19 to enter and wreak havoc on the body. To make this easier to understand, think of it as a city or a town. When the police of the city or town are busy protecting the city from persistent intruders, also known as lung cancer, it leaves the city open to other intruders, also known as COVID-19. So more intruders enter and attack the city, creating a whole catastrophe. Here's a table of possible vaccines, drug possibilities of drug designs in the future. For example, since AstraZeneca targets the COVID-19 virus, the drug could be combined with other lung cancer treatments in some ways to help fight both diseases. I would like to thank my mother and my mentor for this project. For my mother's hospital stories about her lung cancer patients with COVID-19 served part of the inspiration for our project. Thank you so much for listening.